Hello everyone, my name is Ethan Ayler, and today I'm going to be talking about impacts of alcohol use on the brain. Are you looking to unwind during this weekend? Maybe after a heavy week of schoolwork or data analysis, you just pour yourself a little glass of whatever. Maybe you like pina coladas and getting caught in the rain. Maybe you're wasted again in Margaritaville searching for your lost shaker of salt. Or maybe you're sipping on gin and juice.
Laid back.
However, do you ever wonder what alcohol is doing to your brain? Today I'm about to share some information that may make you consider declining Keith from Research Lab's request to go drinking. First, I want to discuss an important mechanism that prevents certain toxins from entering the brain: the blood-brain barrier. The blood-brain barrier prevents direct contact between circulating blood and nervous tissue in the brain. The blood-brain barrier delivers blood to the brain without contact, like me DoorDashing Taco Bell. However, ethanol can pass through the blood-brain barrier, inviting your brain to the party. Urbaniak and other researchers set out to look at the impacts of immediate alcohol consumption on the brain. Their study used proton magnetic resonance spectroscopy and diffusion-weighted imaging. Their study looked at ethanol in the brain, metabolic changes, and microstructural differences with immediate alcohol consumption. Their study collected data from 44 men. In the study, they excluded women and those with alcohol problems. They excluded women in the study because of potential health risks with alcohol consumption. They excluded people with alcohol problems because they only wanted to look at moderate drinking. Participants had to abstain from drinking before participating in the study. The study had 4 sessions. The participant first went through T2 weighted imaging, then they went through proton magnetic resonance spectroscopy and diffusion weighted imaging. After the first session, the participant took 150 milliliters of pure vodka outside the scanner. After 30 minutes of alcohol consumption, the participant went to the T2 weighted imaging, the proton magnetic resonance spectroscopy, and the diffusion weighted imaging. And this repeated 1 hour after alcohol consumption and 2 hours after alcohol consumption. The research found a significant increase in ethanol 30 minutes after consumption, followed by a significant decline of ethanol 1 and 2 hours after alcohol consumption. The study emphasized a heavy ethanol presence in the frontal cortex area. The study also found a significant increase in lipid and lactate levels identified after approximately 1 hour from consuming of 60 milliliters of pure alcohol. The presence of lipid in the brain typically reflects either damage to cell membrane or necrosis. In a healthy individual, a peak of lipids is not visible in a 1-HMR spectrum. However, this study finds a general increase after 1 hour of consumption is quite concerning. Similar to lipid levels, a peak corresponding to lactates is not visible in 1-HMR spectrum of a healthy brain. Visible lactate peaks can provide evidence of anaerobic metabolism and oxygen deficiency due to pathological changes. Visible lactate peaks has also been shown in instant strokes. Their findings also indicate that ethanol stays in the brain longer than any detectable tests. Not only does ethanol stay in the brain longer, it also disrupts inhibitory control processes. A study by Harper and other researchers point to areas like the medial frontal cortex and the dorsal medial/lateral prefrontal cortex is related to successful inhibition. Specifically, they argued that theta, which is 4 to 8 hertz, the power range, is associated with inhibitory and cognitive control processes. Theta was also linked to the communication between the medial frontal cortex and prefrontal regions. Harper and colleagues tried to evaluate a potential causal relationship between drinking effects on EEG and response inhibition. The author of the study also hypothesized that heavy drinking would decrease inhibition-related theta power and have a greater impact on female participants. They also predicted an increase in dorsal prefrontal cortex and medial frontal cortex theta would signal the need for inhibition. The study analyzed the data of 300 twins, 185 women, with 182 monozygotic, with 86 complete pairs, and 118 dizygotic twins with 51 complete pairs. Participants had to complete the Substance Abuse Module, SAM, of the Composite International Diagnostic Interview. The study also collected behavioral and EEG data of the participants using the Wun-Back Go/No-Go task. The way the task works is that 2 white letters would appear on a black background, and participants were instructed to make a button press whenever a letter followed a different letter. For an example, if the second letter was sequenced XY, you would press the button because of the Y. Participants would have to withhold their response when the letter was identical to the preceding letter. For an example, in the no-go, 3 letters in the sequence XYY. The study analyzed the EEG data using time frequency analysis. Before I continue, I want to explain a bit about EEG or electroencephalography to help people understand more about what is happening in this. Electroencephalography, or EEG, collects the electrical activity in the brain. The brainwave activity presents itself in the form of a wave, and this is measured in hertz. There are different types of EEG analysis. Frequency analysis separates the brain's activity into frequencies, like the aforementioned theta. Now, event-related potentiation, ERP, averages the brainwave frequencies, typically during a time-locked task. Now, time frequency analysis is like the child of event-related potentiation and frequency analysis. So what time frequency analysis does is it looks at the average brain activity of a time-locked event depending on frequency range. The study found lower accuracy during the no-go condition compared to the go condition. Drinking did not relate to the error rates in any of the conditions. The no-go trial were associated with greater theta power in the medial frontal cortex. The no-go trial was also associated with greater theta connectivity between the medial frontal cortex and dorsal prefrontal cortex. The study also found that a negative association between the use of alcohol and no-go theta activity was stronger for females. No significant effects were found regarding the go task theta activity or pre-existing genetic/shared environmental risks towards drinking. Basically, what all this means is that normative levels of alcohol use during emerging adulthood has potential sex-specific causal effects on response inhibition. Here is a summary of the key points discussed throughout the video. Ethanol enters the blood-brain barrier. Ethanol generally increases within 30 minutes of consumption in the frontal cortex. After 30 minutes, ethanol levels in the brain decrease. With ethanol, there is also an increase of lipids and lactates in the brain, detectable in 1-HMR. Now, visible lipid peaks are linked with cell membrane damage and necrosis. Oxygen deprivation and anaerobic metabolism is also linked with visible peaks of lactates in the brain. Alcohol also disrupts inhibitory control processes. This effect was especially notable with women. The results of both articles highlight the impact of drinking during young adulthood on the brain. In conclusion, maybe you need to decline Keith's request on going out drinking and maybe decline that second margarita.